My name is Selby, and today I will be discussing the effect of targeted metabolic modulation during in vitro differentiation on the enhancement of the structural and functional maturation of mammalian Müller Glia-Derived photoreceptors. Receptors are specialized cells at the back of the retina that detect and convert light into electrical signals the brain can process. This is known as phototransduction. The outer segment of the photoreceptor is often considered the most integral part of this process, as it contains the light-sensitive protein rhodopsin. Without rhodopsin, phototransduction would not be able to occur. A few layers away from the photoreceptors in the retina are Müller glial cells. They play an important structural and functional role in the retina, and in some studies have shown regenerative properties, differentiating into photoreceptor cells. Research is to determine if metabolic modulation of Müller glial cells would induce differentiation to photoreceptor cells. This was determined by the presence of rhodopsin, as it suggests Müller glial cells have differentiated into mature photoreceptors containing outer rods. First, I'd like to thank the Sarasota Community Foundation for providing a generous grant to allow this project to be possible. I would also like to thank Lions World Vision Institute, specifically Dr. Foster, for providing lab space, some materials, and assistance throughout experimentation. Dr. Foster oversaw my experiment and demonstrated how to perform various steps so I could accurately conduct my research. Go on to discuss my methodology. Following thawing the cells, they were resuspended in a T25 flask containing fresh culture medium and incubated to allow growth and attachment. After 2 days of incubation, the cells were checked under an inverted microscope to confirm morphology and adhesion, and once adequately confluent, trypsin EDTA was added to allow for cell detachment. Following cell detachment, the trypsin was neutralized and equal portions of volume were transferred to each well containing a poly-D-lysine-coated cover slip. Cells were then incubated for 24 hours with complete medium to prevent excessive stress. The following day, the old complete medium was replaced with serum-free differentiation medium and incubated overnight. Every 2 days, the medium was replaced. After 10 days of differentiation, the cells were incubated in 4% of PFA for 20 minutes at room temperature. After rinsing, the cells were incubated in 0.1% of Triton X100 for 15 minutes at room temperature. The rhodopsin antibody was then added to each well and incubated overnight, wrapped in foil to avoid light. The next day, the cells were washed and the secondary antibody was added. Following 1 hour of incubation at room temperature, the secondary antibody was replaced with Alexaflor 594 and left wrapped for 15 minutes. Cells were then plated on slides and visualized using a fluorescence microscope. This methodology was relatively strong, as the overall process of cell passaging and differentiation is well published. However, due to limited available knowledge of the effect of metabolic modulation on differentiation, specifically regarding Müller glial cells, I was unable to definitively determine if it would encourage differentiation. On to the real-world impact of this research. Currently, there is more than 2.2 billion people suffering from vision impairment in the world. Around 1 billion of these cases could be prevented or have yet to be treated. Some of these diseases that cause said vision impairment include age-related macular degeneration, diabetic retinopathy, and more, such as retinitis pigmentosa and cone rod dystrophy. All of these diseases have one thing in common, and that is photoreceptor death. Photoreceptors are very fragile cells due to various reasons, such as their high energy demands and their highly specialized structure. Once they have died, they cannot be replaced, which leaves millions of people blind and sight impaired. The project aims to provide a pathway to restore this vision through the replacement of photoreceptor cells. The results found did not support the hypothesis that medium concentration of lactate and glucose would encourage differentiation, nor did it support any other concentration group inducing differentiation. While no mature photoreceptors were shown through visual analysis, due to a technical error resulting in a lack of testable cells, further evaluation through PCR testing could not be done. Essentially, this means that it was unable to be known whether the cells had partially differentiated. One unexpected finding of the experiment was the beneficial impact of increased lactate added to the Müller glial cells. High concentrations of lactate reduced stress and increased the health of the Müller glial cells, whereas low glucose induced extreme stress, causing central necrosis and NSAID aggregation.